right, so let's begin by going over some of the bones again. Let's start with the skull. So we're looking at a frontal view of the skull. So our forehead, that's the frontal bone. The ridge of bone above the orbits is the supraorbital ridge. So remember, supra means above, and the orbits are the eye sockets. So supraorbital ridge. Nose bones are nasal. Our cheekbones, those are called zygomatic bones. The opening where our nose will go, that's the nasal aperture. Okay, so as you notice in the forensics lab, some of the exercises are asking you about measuring the nasal aperture, which obviously isn't possible, you know, if you're looking at a picture, not an actual skull. And then this bone right underneath the nasal aperture, where the upper teeth attach, this is the mandible, and then this row of teeth is called the mandibular teeth. Okay, so from the front view, from an anterior view, we see the frontal bone, we see the supraorbital ridge. We can see the orbits, the eye sockets. We can see the nasal bones. We can see the nasal aperture, the opening. We can see the zygomatic bones, the cheekbones, and we can see the maxilla. Okay, now we're going to turn the skull to the side. So now we're looking at a lateral view. So from the lateral view, we can still see the nasal bones. We can see the maxilla. We can see the zygomatic bones, the cheekbones. This bone by your ears is the temporal bone. So think temple, temporal. This is the temporal bone. And then superior to, so remember superior means above, so directly superior to the temporal, we have the parietal. And then also you can see part of the frontal bone as well. Okay, so temporal is here, superior to it, we see the parietal and the frontal. All right, right behind your ears, everybody feel that bump behind your ears? That's called the mastoid process. Mastoid process. Turn the skull around and look at it from the back, from a posterior view. So this bone here in the back of our skull, that's the occipital. And then superior to the occipital, we have the two parietals. Okay, so if the exercise is asking about a bone that is anterior to the occipital, just means in front of. So you could say that the frontal bone is anterior to, you could say that the nasal bones are, you could say that the mandible is, or excuse me, the maxilla is. So from the back, we've got the occipital and the parietals. Okay, now looking at a superior view or a view from above. So we're looking at the skull from above. We can see the frontal bone and we can see the two parietals here. Okay. All right, so then the jawbone, the mandible. So this entire structure is called the mandible. This part of it is the ramus. This part here, your chin, that's something that's unique to anatomically modern Homo sapiens. We have a protruding mandibular symphysis. That's our chin. Okay. All right. So just to give you guys a little preview. When we get to the human evolution sections, the reason it's so important to learn about these bones now, this is one of our human ancestors here. So obviously when you flip the skull over, you see it sinks in quite a bit behind the supraorbital ridge. That's called the postorbital constriction. So that means that this hominin has a relatively smaller brain size in comparison to modern humans, especially the frontal region. If you notice the zygomatic bones, the cheekbones are jutting, excuse me, are jutting way out to the side. So that's why it's important that we're learning about zygomatic bones now and how to identify them. When you look at the supraorbital ridge, it's double arched and far more pronounced. Also, when you look at the mandible, this is Australopithecus boisei or Paranthropus boisei. When you look at the mandible, it's huge in comparison. So this is a modern Homo sapien. This is Paranthropus boisei. So you can see the size difference. And then also looking at the dental arcade, you can see that boisei has more of a rectangular dental arcade, whereas modern Homo sapiens, we have a more parabolic or rainbow-shaped dental arcade. And also look at the size of the teeth as well. So boisei ate a diet of mainly rough, tough vegetation, required really tough jaw muscles and chewing muscles. So that's why they have really huge dentition, because they're crushing up vegetation. Whereas modern Homo sapiens, we have a much more omnivorous diet, and we're also using fire and utilizing tools to break up our food before we consume it. All right, so when we flip the jaw this way, we can also see the dental formula. So when we're counting dental formula, we start at the front of the mouth. We're starting with the anterior dentition. So in the front, we can count on each side 2 incisors, 1 canine, 2 premolars, and 3 molars. So that's where we get the dental formula 2, 1, 2, 3. And if you've had your wisdom teeth removed, then that makes your dental formula 2, 1, 2, 2. Okay? All right. So the other 2 parts of the skeleton I really want you guys to focus on is the femur, the thigh bone, and also your pelvis. So your femur, is going to articulate. So remember that word articulate just means touch. So here's our pelvic girdle here. So at the proximal end, so remember proximal is closer to the connection to the body. So at the proximal end, the head of the femur will articulate with the acetabellum of the pelvis, or more specifically of the ischium. Okay, so that's the first place where it articulates. At the proximal end, the head of the femur articulates with the acetabellum of the pelvis. Okay, and then down at the distal end, so down at the distal end, the femur is going to articulate or touch with the medial and lateral condyles of the tibia, of your shin bone, and it also articulates with the patella. Okay, that's the kneecap. So there's a question that's asking about what bones does the femur articulate with or touch. So your answer would be, if you're just talking about the whole bone, it would be the ischium of the pelvis, or if you wanted to be more specific, you could say the acetabellum. And then at the distal end, it's articulating with the lateral and medial condyles of the tibia, and also with the patella. Okay, and then looking just at the femur, we're looking just at the thigh bone now, and we're looking at an anterior view, so a view from the front. So if we know that the head of the femur articulates with the acetabellum, and we have it oriented in the right direction, then we know this is a right femur, okay? And we know that the front side is very smooth. Down here at the distal end, we have the condyles, the lateral and medial condyles. Up here at the proximal end, you can see this big protrusion here. This is the greater trochanter, and you can kind of see the lesser trochanter poking through over here. So larger bump is the greater trochanter, smaller bump is the lesser. This here is just called the head of the femur. That's where it articulates with the acetabellum of the pelvis. Flip the femur around, so now we're looking at a posterior view, a view from the back. So this ridge, this protrusion along the back, this is going to be really important when we start learning about bipedalism. But right now we're going to learn that this ridge is called the linea aspera. Okay, all right, we're looking at so we're still at the posterior view, but we're looking at the proximal end, the end that's closer to the pelvis. So we can see the lesser trochanter much better here, and we can also see the greater trochanter, lesser trochanter. Other adaptation that's going to be important is the head of the neck of the femur, excuse me, so the head of the femur here. This is the neck of the femur. So in a biped, there's going to be much more of an angle. In a quadruped, like a chimp, it's going to be much straighter. Okay, so we're going all the way down now to the distal end. So this is we're still looking at the back of the femur, but this is the end that connects with the tibia. So we can see on the back side our condyles, our lateral and medial condyles. Okay, and then pelvis. So the entire structure, looking at it all together like this, and again, envision that the sacrum is in the back. I just don't have enough hands. But this entire structure is called the pelvic girdle, including the sacrum and coccyx in the back. So each half is called either a nominate or os coxa. Okay, and this is made up of 3 fused bones. So the top half is called the ilium, bottom half is called the ischium, and then where it articulates with the other nominate bone, that's the pubis bone there. Okay. In the pubic symphysis, there's cartilage that would go in between there. I mean, obviously this is just the bone replica, but there would be cartilage that goes in between there called the pubic symphysis. Okay. All right, so this entire bone, either innominate or os coxa, top half ilium, bottom half ischium, and then where it articulates with the other innominate, that's the pubis. Okay, and then where it articulates with the femur, this ball socket joint, that's called the ossa tabellum. The ridge, this kind of crest, so if you're standing up and you have your hands on your hip, so that crest that you're feeling at the top of your hips, this is the iliac crest. So remember, this is the ilium, and then the crest is called the iliac crest. All right, so in the forensics lab, it does have you do one activity where you're identifying biological male versus biological female. So it's going to have you look at pictures of two different pelvic girdles and have you determine which one's male and which one's female. So the features that you're looking for, so when you turn the pelvis around and look at it from a lateral view, you can see this little notch here. So this notch is called the greater sciatic notch. So easiest way to tell, obviously in your lab manual, it's not going to be super easy to do this, but I just want to show you guys if it's difficult to fit your wrist in the greater sciatic notch, see how there's that opening there? If it's difficult to fit your wrist in there, it's likely a male, and this is a male pelvis. Because a male has a narrow greater sciatic notch. If this were a female, the greater sciatic notch would be wider, which is pulling this part back. So this is where the ilium would articulate with the sacrum. Let me pull up the sacrum. Where'd our sacrum go? All right. So this is where the ilium articulates with the sacrum. So if that greater sciatic notch is wider, then what it does is it actually pulls back the sacrum and the coccyx, which is going to make childbirth much easier. Okay, so that's the purpose of a wider, greater sciatic notch in a female. Other features you're going to look for in this lab, in lab 7. So this angle here, that's the subpubic angle. So it is relatively narrow in a male, and it's going to be wider in a female, which is again opening up the birth canal to make childbirth more pleasant. All right, and then when we flip it up like this, so now we're looking at a superior view. So again, try to envision that the sacrum is where my head is right now. So when we flip it this way, looking at a superior view, this is a male pelvis, so this is relatively narrow, but in a female, it's going to be much wider. Okay? So I do want you guys to do that lab exercise, because I do believe it is possible to determine male versus female looking at those 3 features with the pictures. And, oh, so I'm just going to do a few corrections on some of your guys' lab exercises that I'm seeing come through. So let me bring it up. Okay, so now we're talking about our bones that are in our arm. So in your upper arm, this is your humerus. This entire bone is called the humerus. So at the proximal end, it's going to articulate with the scapula, okay, with the glenoid fossa of the scapula. So we've got our scapula, the humerus articulates this area here of the scapula, of the shoulder blade is called the glenoid fossa. So the head of the humerus articulates with the glenoid fossa of the scapula. At the proximal end, down at the distal end, so we've got the oscillarian fossa here, oscillarian fossa, it articulates with both the radius and the ulna. Okay, so at the proximal end of the ulna, so proximal end is where it will articulate, where it touches with the humerus. So at the proximal end, we have this U shape that's called the oscillarian process. Okay, so that's at the proximal end of the ulna. And then down at the distal end, we've got the styloid process. Okay, so there's a question that's asking about the bump on your wrist. So remember, we're always thinking about it in anatomical position. So our wrist is out like this. So on the right side, another way to remember it too, on the right side is your radius. And then on the side that's closer to your body, if you're in an anatomical position, that's the ulna. Okay, so on this end, at the proximal end, you're feeling the styloid process. The styloid process of the radius on the right side, and the styloid process of the ulna on the side that's closer to your midline, closer to your body. Okay, so that's the styloid process on both sides. Bones in your hand. So bones in our wrists, those are called carpal bones. So the way I remember it, if you have carpal tunnel, then your wrist hurt. So carpal tunnel, those are the bones in our wrist. The bones in our hand, those are metacarpal. Bones in our hands are metacarpal. The first row of phalanges, so remember both finger bones and toe bones are called phalanges. So the row of phalanges that are closer to the metacarpals, those are the proximal phalanges. The middle row, those are the intermediate phalanges. And then the third row, essentially our fingertips, those are the distal phalanges. Okay, so our thumb only has, so there's the metacarpal on the thumb, and thumb only has 2 rows of phalanges. The thumb has the proximal phalange and the distal phalange. That's another question in your lab manual as well. Okay, so all the other fingers, your index, middle, ring finger, and pinky have 3 rows of phalanges. The thumb only has 2. Okay, other feature of anatomically modern Homo sapiens and our ancestors, we have an opposable hallux. Or actually, hallux is a big toe. We have an opposable big thumb. We'll get to hallux when we get to the feet. Okay, and that's something that we share with the other primates. Other primates also have an opposable thumb. Our thumb is much shorter and stronger. That is allowed for production of tools. All right, bones in the hand. The other one I was going to do. Oh. Okay, so on one of your lab exercises, it's asking you about the bumps that you feel on your ankle. All right, so your tibia is your shin bone. So this bump here, that's the tibial tuberosity. So when you feel right underneath your kneecap, that bump you feel is actually your tibia, that's the tibial tuberosity. And then when you go down all the way to the distal end, so where it connects with your foot, where it connects with your ankle, on the inside of your ankle, what you're feeling when you feel that bump on the inside of your ankle, you're actually feeling the distal end of the tibia, which is the medial malleolus. Okay, medial malleolus. And then the fibula, when you feel that bump, so the fibula is lateral to, it's a long skinny bone that is lateral to the tibia. So what you're actually feeling when you feel that bump on the outside of your ankle, so when you're going down your ankle, your leg, and you feel the outside, but you're actually feeling that bump is the distal end of the fibula, which is called the lateral malleolus. Which if you think about it, it makes sense, because I mean, lateral means more to the side, and medial means more towards the midline. So the inside of your ankle, what you're actually feeling is the distal end of the tibia, which is the medial malleolus. And then on the outside of your ankle, what you're feeling is the lateral malleolus, which is the distal end of the fibula. Okay? All right. Let me show you how it articulates too. All right. So the tibia is going to articulate, is going to touch the talus. So the talus is part of your ankle. So this bone here is the talus. It articulates with the distal end of the tibia. So we have the talus. The heel bone is called the calcaneus. Heel bone is calcaneus. Bones in our ankles, those are the tarsal bones, tarsal bones. Bones in our foot, these are metatarsals, metatarsals. The first row of phalanges, remember both toe bones and finger bones are called phalanges. So the first row are the proximal phalanges. The intermediate row is called the intermediate phalanges. And then your toe tips, the very tips of your toes, those are called the distal phalanges. Okay, so just like your thumb, your big toe, your hallux, so in humans and bipedal hominins, we have a hallux, a big toe that's in line with the rest of our toes. We'll learn in some of the upcoming chapters that our living relatives and our ancestors have an opposable hallux, so one that would orient out to the side like your thumb. So we have the metatarsal here in our business. We're looking at the big toe. We have the metatarsal, but we only have 2 rows of phalanges in the big toe. So we have the proximal row of phalanges and the distal. Okay? All right, how you guys doing so far?
Doing good.
Is this helping? You guys have questions?
I have a question.
I get it.
I have more.
Okay.
Yeah, I have a question more about the lab exercises. I mean, the another
Okay,
concept. Sorry.
the concept review questions.
Um, so is the lab 7 concept questions due today?
No, Lab 7 concept review questions are due by next Friday, which I believe is the 16th. I think originally this unit was supposed to end on the 14th, but I wanted to give you guys a couple more days. I posted an announcement yesterday morning, I believe. So I'm giving you guys a couple more days with lab number 5 and 6. For lab number 7, the concept review questions are still required. And I want you to do 2 of the exercises for 7. I want you to do the one that's related to sexing the pelvis, determining biological male versus female. And I would like you to do the exercise that's determining cause of death. And I'm going to go in and label it for you guys. I'm going to label what's required. But I have to leave them all in there, assuming you guys want the extra credit option. I have to leave them in there, so I'll label the ones that are required, and the ones that are extra credit. Does that sound like a fair deal?
Yeah, thank you.
Yeah, of course. But yeah, definitely make sure you've done the concept review questions for 7, because there will still be some questions on your next exam that are related to forensics. Obviously, I will remove any of the ones that we didn't focus on, but you should still go through. And I'm going to go through the PowerPoint with you today. You should still go through the PowerPoint, and you should still do the concept review questions for Lab 7, which is forensics. Okay, good question. Other questions? Okay. All right, so you guys are you feeling a little bit better about the human skeleton and the parts I want you to focus on, the parts of the skull, femur, and pelvic girdle? All right, so we're going to go here to share screen. All right. Are you guys able to see my screen?
Yes.
All right. So lab number 7, so this is the PowerPoint that goes along with lab number 7. So this is going to be focusing on forensics. So forensic anthropologists are specialized in determining cause of death. They are specialized in determining, if you have a huge assemblage like this, determining how many individuals are in that assemblage, that fossil assemblage, or that assemblage of, possibly modern humans that are in a mass grave like that, determining how many individuals are actually present there. They're also specialized in determining the biological sex and the age of those organisms. If you have a huge assemblage like that, a mass grave, wanting to determine how many males are in that population, how many females, how many juveniles, how many adolescents, how many adults, how many elderly. That's what they would specialize in determining. So they're essentially creating a biological profile, identifying victims, and determining and reconstructing the events that may have surrounded their death. This can be done for humanitarian reasons. If you're trying to understand, for example, if there was a major natural disaster in a certain region and we wanted to determine exactly how many humans were affected by it. Humanitarian reasons could be a motivator for being a forensic scientist. Criminal cases, obviously determining, if there's a body found, we have to first, of course, determine who that individual might have been, how old they were, if they were male or female, what their ancestry might have been, and their cause of death. Also mass fatalities, war crimes, natural disasters. These are all areas that forensic scientists really specialize in. Forensic scientists were used to help excavate and understand the remains after 9/11. Of course, once the first responders went in and made sure that anybody that was still alive was rescued, forensic scientists were the next step that wanted to help us better understand what happened. All right, bioarchaeology is related to forensics. Bioarchaeology is the study of the human skeleton by looking at archaeological sites. So forensics is more modern, and then bioarchaeology is looking at more historical sites. So bioarchaeology emphasizes approaches from biological anthropology and archaeology. That's why it kind of seems like a hybridization of those two words, and derives theoretical guidance from both cultural anthropology and evolutionary biology. Bioarchaeologists use the methods of skeletal biology, mortuary archaeology, so looking at mass graves and cemeteries, and archaeological records to answer questions about lives and lifestyles of past populations. So forensic anthropology and bioarchaeology are both subfields that go underneath the grander scope of biological anthropology. So they're even more specified than the 4 branches. So the 4 branches, if you remember from chapter 1, biological anthropology, cultural anthropology, archaeology, and linguistics. So forensics and bioarchaeology go underneath the overarching category of biological. Both fields are going to rely on skeletal analysis to gain information about humans, both past and present. Both fields are multidisciplinary, meaning that we're going to draw in information from the other sciences, from anatomy, from biology, even chemistry and forensics, when we're chemistry and physics, when we're learning about dating these materials, we have to have a knowledge of chemistry. Both apply the same methods and techniques and follow the same protocol steps. So whether you're a forensic anthropologist or a bioarchaeologist, you're going to follow the same protocol. So why study bones? So bones can survive the process of decay. So when you're looking at fossil remains, for example, you don't necessarily have all of the nose because the nose is made mainly of cartilage. So all that's left there is that nasal aperture, that opening. But the bone itself preserves quite well. Bone is mostly calcium phosphate and other ingredients such as collagen, fiber, and crystalline salts, and hydroxyapatite salts. So this is just one of the reasons why bones are generally, they generally preserve fairly well in the fossil record. All right, so forensic anthropology objectives, obviously recovering skeletal remains of past humans and more modern humans, skeletal examination, forensic identification, so identifying their age, biological sex, time of death, things like that, reconstructing identity. So there's steps that we'll go through. So of course, when we discover bones, we want to first determine, are they human bones? Are they from some other kind of animal? I have a colleague at Ventura College that used to work for the FBI, and she tells me she still has police officers come in and bring her bones and ask her if they're human or not. So that's the number one thing. You're trying to determine, are these bones humans? Are these from a human? How many individuals are represented if it's a mass burial kind of situation? How long ago did death occur? Did these humans recently die, or have they been dead for years, decades, hundreds of years, thousands of years, millions of years? What was the person's age when they died? Were they juvenile? Were they young adult? Were they older adult? Were they elderly? What was the person's sex versus biological male or female? What was the person's ancestry? Now this one is tricky, and I've had forensic anthropologists tell me different things about this. It is very important not to confuse ancestry with race. Ancestry is quite simply talking about, geographically speaking, where did our ancestors originate, which doesn't have anything to do with race. We're going to learn that race is actually a biologically invalid category. There's no genetic or skeletal or forensic evidence really to support race. Ancestry is just talking about geographically, predominantly, where did your ancestors originate? So it's really just dividing it into European ancestry, African ancestry, Asian ancestry, Native American ancestry, Australian ancestry, things like that. But obviously, as modern humans, we are continuing to engage in gene flow. So if you have European ancestry, you may certainly end up having offspring with somebody with African ancestry. Or if you have Asian ancestry, you might end up having children with somebody with European ancestry. You guys get the point. So, the more genes continue to flow, and the more that, modern Homo sapiens are very genetically similar. There are definitely some aspects of the skeleton that can help us determine ancestry with, some level of certainty. Like, for example, this skull, this is an African female of African ancestry. But it's difficult to determine ancestry unless you have the actual skull and you're a specialist. So that's why I'm not going to require you guys to do the activity that's related to ancestry. I mean, of course, if you want to give it a shot for extra credit, I absolutely encourage you to do so. All right, number 7, so looking at how tall the person is. So the bone that is going to be critical for this is the femur. So there's an equation that you can use. You take a measurement of the femur, and then you're going to plug in that measurement into this formula to determine how tall that person would have been within, it's not going to be necessarily 100% accurate, but it's within a couple inches. All right, also number 9 is determining how that person died. Was it a violent reason? Was it blunt force trauma? Was it, was there some sort of stab wound, a gunshot wound? Were they hit with a blunt object? Did they have some sort of degenerative disease that caused them to die? So these are all things that leave their marks on the bone. Another thing that leaves their mark on their bone is nutritional factors. So if you suffer from nutritional deficiencies early on in life, it leaves the mark on your bones and also on your teeth. There's also something called porotic hyperostosis that is found in some early agricultural populations that indicates iron deficiencies. Because when we first started practicing agriculture, we were focused in on eating just certain food types, just one type of wheat or one type of barley or corn or rice. So you can imagine if you just eat carbs, if you just eat wheat or rice, you may end up with an iron deficiency. So that really explains why the early agriculturalists didn't necessarily have it all figured out. They need a more varied, diverse diet. All right, so some of the standard techniques. So techniques developed in physical anthropology and archaeology of how to excavate a site with multiple individuals, or sites that just have 1 or 2 individuals. Also proper excavation techniques. Obviously you don't want to damage the skeletal materials as you're going through this process. Also the set of legal guidelines. So you have to think about, if these bones, for example, are ancestors to the Chumash, you obviously would want to make sure that you have proper and adequate permission from the Chumash before you would go through and excavate that site. You don't want to be a grave robber, essentially. Also documentation, measurements, sketching, and photography. So, knowing what bones to measure and how to measure them and what this tells you. So for example, measuring the femur is going to give you information about stature. Measuring parts of the skull might give you information about ancestry. Measuring the pelvis is going to give you information about biological male versus female. Measuring nasal aperture and the eye sockets is going to also give you some information about the age and sex of the individual and possibly ancestry. It utilizes a multidisciplinary approach of collaboration with forensic pathologists. So pathologists look more at how disease affects bones, odontologists, which study teeth, and also homicide investigators, because, of course, a homicide investigator may not have the expertise as far as how to identify the bones and determine cause of death, but they certainly have a stake in this whole investigation, especially if they're in the middle of investigating an active crime scene. They certainly want to know as soon as possible, how to identify that person and determine how they died. Also to identify a descendant, or an ancestor or relative, point out evidence of foul play, and assist with time of death and death estimates. All right, so excavated remains are cleaned in forensic labs. So you're collecting the bones, obviously taking great care. You're collecting the bones in a specified manner, and then once they're brought back to the lab, that's when they become cleaned. Because obviously you don't want to try to do something like that out in the field when circumstances are out of your control. You want to try to do this when you're in a lab in a controlled environment. So once they've been cleaned, then they're arranged in anatomical order, which it totally bums me out we can't be in the lab room, because I actually have you guys organize the disarticulated skeletons in anatomical order just like this, which is really fun to do. It's putting all the pieces together. So I'm bummed out that we can't do this semester, but you're putting that skeleton together, a disarticulated skeleton together in anatomical correct position. Anthropometry, so that word there, anthropometry, is measurements of human proportions. It's a quantitative method, so think quantitative. You're taking specific measurements. You're actually measuring the long bones. You're measuring, there's measurements that they take of cranial width. So at the parietals, that's the widest point for modern humans. They take measurements of cranial width and also cranial length. So going all the way from the supraorbital ridge back here to the occipital. Taking measurements of the pelvis, taking measurements of the head of the femur, the head of the humerus, for example. So that's anthropometry. And then osteometry, that's measurement. So osteometry is measurements of the bone. Anthropometry is measurements of still living humans. Biological profile. So if you had this as a forensic anthropologist, if you had this skeleton laid out in your lab, once you have the bones cleaned and arranged in anatomical order, one of your first priorities is to develop a biological profile. So you want to determine, are you looking at a male or a female? And you want to determine, and sorry here, we should erase race, that's an old term. Instead of race there, pretend that says ancestry. Ancestry, but again, that's difficult to determine. We're just looking at, is it more likely that this person's ancestors originated in Europe, Africa, Asia, the Middle East, so on and so forth. So we're looking for ancestry, age at time of death, but you're not necessarily going to be able to determine the exact age. You won't be able to determine, okay, this was a Caucasian female that was 23 when she died. It's going to be more of an age range, such as infant, juvenile, adolescent, young adult, older adult, elderly. Age and stature. So stature, that's another name for height. So the easiest way to determine stature, if you have a femur, is you're going to use that equation. So you take a measurement of the femur that goes all the way from the head of the femur all the way down to the condyles. So you're measuring from the head all the way down to the condyles using a tool called an osteoboard, and then utilizing that equation to determine how tall that person was. And postmortem interval. So essentially, how long has it been since this person died? How long ago did this person die? Some additional assessments, you're looking for any sort of trauma. So sometimes it's trauma that may have healed. So for example, maybe this person broke their femur 10 years ago, but they didn't die from it. You'll be able to determine that because you'll be able to see whether the bone is healed or if it's not healed. If that trauma is not healed, then it's likely that person died from that trauma. But if that trauma is healed, then they likely survived and lived another day. Pathology, so looking for any disease or nutritional deficiencies. Workload, as some of you in my lecture class are learning about here in chapter 5 in lecture, we talk about the wolf hypothesis. So bone is essentially created. Bone becomes more robust and denser for populations that are more active. So if you have a high level of workload, then your bones are going to be denser. If you're looking at a population or a human that's more sedentary, maybe a human that worked a desk job for 40 years, they may end up having more gracile skeletal anatomy. Also diet. So looking at dentition can determine diet. Also looking at, the bones can tell you about the nutritional capacity of that human. All right, so this slide here, so this one will be important because we are going to still do the activity that's determining male versus female. So in this picture here, so you have the top pelvis, the top pelvic girdle is a male pelvis, the bottom one is a female. So in this picture here, you're looking at the superior view, the view from above. So, and it's nice here, you can see that the sacrum is attached in the back. I don't have enough hands to attach them when I show you guys the bones. So that you can see the sacrum in the back here. And you can see how for the female, that pelvic inlet, essentially the birth canal, is much wider in the female, much, much wider in comparison to the male. And the reason for that is the greater sciatic notch. So now envision that you're looking at the side of the pelvis. So that greater sciatic notch, if it is wide like it is in the female, is going to pull back. That greater sciatic notch, if it's wide, will pull back the sacrum and coccyx, which will open up that pelvic inlet, okay? All right, you can also use the skull to determine male versus female. However, and I believe it's really difficult to determine by looking at pictures, so I'm not going to make you guys do that. But just so you know, there are some features of the skull that will determine male versus female. The supraorbital ridges, so again, remember that terminology is coming back. That terminology that we learned in 5 and 6 is coming back. So the supraorbital ridge, the brow ridges, are going to be more prominent in males, less prominent in females. The frontal bone is going to be steeper and more rugged in a male. It's going to be rounder and smoother in a female. The zygomatic arches, the cheekbones, are going to be extended past the external auditory medius in a male. So you see here, the external auditory medius, that's the hole where the ears go. So in this one, this is a female. So the zygomatic bones do not extend past the external auditory medius. The orbits in a male are going to be thicker and more blunt, and in the female, they're going to be sharper and rounder. So you can kind of see that in this female here. They're more rounded than you would see in a male. I wish I had a male to show you guys, but the skull is from a female and the pelvis I have is from a male. All right. So determining age. So immature or juvenile versus a mature or adult skeleton. So before puberty, biological identification of remains can be difficult. So if that human has not gone through adolescence, it's going to be really difficult to determine whether it was a boy, a male child, or a female child. The bones are mostly cartilage. They don't preserve as well. Growth plates are not fully fused. Sexual differences, or what we call sexual dimorphism, is not nearly as pronounced until that human reaches adolescence. And the best determinant, if you're looking at a juvenile, would be looking at a skull with dentition, because you can look at the eruption rates to determine how old that infant or child might have been at the time of death. Other ways that you can do age determination if you're looking at a human that's past adolescence. So formative changes, those are changes that we go through as we're growing during our developmental stages. And degenerative changes, those are changes that we go through as we're aging, as we're getting older, as we're going through the senescence process. That's the aging process. Dentition, so looking at what we call eruption rates, so we can determine if we're looking at an infant, toddler, juvenile, we can look at the eruption rates of dentition to determine the age within a range. We can look at the skull sutures. So the sutures, so like for example, this is the sagittal suture here. The sagittal suture is separating the two parietal bones. We can look at the growth areas of the long bones and the pubic symphysis. The pubic symphysis will start to deteriorate as you get older. So the pubic symphysis, that's where the two pubis bones of your two os coxae, that's where the pubis bones are going to articulate. That's the pubic symphysis there. So you can look at the deterioration rate of the pubic symphysis to determine whether you're looking at a young adult or an older adult. All right, this is the one that in my mind has a lot of controversy. Not every forensic anthropologist I have talked to agrees that this is still a valid aspect of forensics because of the reasons I told you. Race and ancestry are kind of a complicated topic. Ancestry is just referring to, geographically speaking, where did your ancestors originate? Are they more of Asian ancestry, European, or African, are the three main categories that forensic anthropologists generally look at. So the easiest way to do it, if you are trying to determine ancestry, is to do two measurements that they call the cranial index and the nasal index. So again, because we're not in the lab room, I'm not going to be able to have you guys do these measurements, but the cranial index is cranial width multiplied by 100 divided by cranial length. So cranial width, you're looking for the widest part of the skull. So for modern humans, that widest part is all the way up here at the parietals. So right now you're looking at a superior view, all the way up at the parietals. So you would use a tool called a sliding caliper, take a measurement of the widest point, multiply that number by 100, and then you're going to divide it by your measurement for cranial length. So you'd use that same caliper, and you're taking a measurement that goes all the way from the supraorbital ridge back here to the occipital. Okay, so that measurement is called cranial length. So your equation for cranial index is cranial breadth multiplied by 100 divided by cranial length. Okay, that's cranial index. And then there's this chart that will tell you, depending upon what your number is, it's going to tell you whether or not that human is more likely Asian, European, or African ancestry. But again, this is extremely controversial. When I have the lab in person, I also have you guys do your own cranial index. And oftentimes you'll find that, for example, when we do my cranial index, I come up as African ancestry, when obviously, I'm more of European mixed ancestry. I do have, when I did my 23andMe, I do have 2% Congolese, but my predominant ancestry, if you're looking at my genetics, is European. However, when you do this cranial index measurement, my ancestry comes up as African. So it is controversial. All right, so if we're determining height, so essentially the stature, how tall somebody was or is. So you're looking here, you're using this wooden board here, that's called an osteoboard. So here they're measuring a femur. So remember, that's the bone in our thigh, and here they're measuring the humerus. So there are equations, and they are in your lab manual if you want to give it a shot. But the equation that you're going to use, if you want to try it for extra credit, you're going to take the measurement that they give you for the femur, and then you're going to take this number, so 2.38 multiplied by the femur length, and then you're going to add 61.41, and that's going to equal the approximate stature with a possible error. So plus and error, that's the plus and minus, that's the error margin, 3.27. Okay, so you're getting pretty close to the stature within a few inches. Okay. All right, so this is one of the areas I do want you guys to give a try. I want you guys, because this is oftentimes the area that students are really interested in anyways, is looking at cause of death and manner of death. So manner of death is essentially what the circumstances surrounding their death. So was it an accident? Was it a suicide? Was it a homicide? Did they die of natural causes? Did they die of undetermined causes? Because sometimes, even a forensic scientist won't necessarily be able to determine without, with no shadow of a doubt exactly how a person died if they died from something that didn't affect the bones. Like say, for example, they were poisoned. That's not necessarily going to come up unless they still have the ability to test what was in that person's digestive tract. If they only have bones left, they may not be able to determine if a person was poisoned. Cause of death is essentially the medical reason. So was it blunt force trauma? Essentially, what caused their heart to stop beating? Was it blunt force trauma? Was it sharp force, like a stabbing? Was it asphyxia? Were they strangled? Did they drown? Was it a gunshot wound or other? All right, so antemortem, so this term antemortem, that means before death. So bone healing or bone remodeling. So if you can see, like say, for example, this individual here has a broken femur, but we know that this person didn't die from that broken femur. I mean, obviously their femur's got some issues going on, but we can see this, we can see how it grew back here. So we can see that this person did not die from their injury. Looking at age and the health of the individual, how old were they when they died? Were they healthy? Did they have good nutrition? The nature and extent of the injury, the location of the injury. Bone healing has a very distinctive look. It's bony callus and also smooth edges. So usually no clues as to cause of death sometimes. If it's, especially in modern day, humans don't usually die from just a broken bone. I mean, it's certainly possible. It's not unheard of. But just because you're able to see some previous fractures doesn't necessarily mean you're going to be able to determine that that was the cause of death. All right, perimortem trauma. This is trauma that happened right around that person's death. So you see the difference here, like in comparison to this previous picture here, where you can see the healing. So looking at these pictures, see how it's much sharper. You're not seeing that soft look to it, that you can tell that bone healed. These are much sharper fractures. So right here, you're looking at the fibula here. You're looking at, oh no, I'm sorry, that's the radius. I'm looking at it from a weird angle. So you're looking at these, very sharp breaks. So these lesions have no signs of healing. And here you're looking at the occipital bone. You're looking at the back end, the posterior view of the skull. All right, postmortem trauma occurred after death. So after the person died, it could have been some sort of trauma that occurred to the body. So this may be marks that are left by animals, or it could be, say, the person died in some sort of environmental catastrophe. Some of these marks may have been made once the person already died. It could be intentional dismemberment, if you're looking at like a serial killer kind of situation, tool marks that are left on bone, or unintentional dismemberment. So these would be animal marks left on bone. All right, pathology. So this is looking at diseases that may have affected the individual. This is looking at nutritional deficiencies, things like rickets, osteoarthritis. So pathological or traumatic processes affect the normal balance between the osteoblast. So remembering back to chapter 6, or I believe it was chapter 5 actually, the osteoblast, that's the bone forming cell, and the osteoclast, the bone destroying cell. So these can be metabolic diseases, degenerative diseases, infectious diseases. Tuberculosis, for example, can cause deformations of the cervical column and of the vertebrae. Syphilis, so syphilis causes these deformations on the cranium, on the skull. So there's different types of syphilis. I mean, of course, I'm sure you're all familiar with the sexually transmitted disease syphilis, but there is also right around the time that we adopted agriculture 10,000 years ago, there was a type of syphilis that was spread in a different way, that was spread more by contact, not necessarily sexual contact. All right, so a fracture through a bone, which has been made weak by some underlying disease, is called a pathological fracture. So if somebody has a broken bone because they have poor nutrition, because they don't have adequate calcium, adequate phosphate in their diet, their fracture may be a pathological fracture. A closed or simple fracture, a break that does not penetrate the skin. So this one here, this is a closed fracture. So is this one as well. Yeah, anything that does not penetrate the skin is closed. And then an open or a compound fracture is a broken bone that penetrates through the skin. And the green stick phrase, these are hard to repair, breaks like a green twig. These bone fractures are treated by reduction of activity, and also immobilization. So putting your arm in a cast, for example, or your leg in a cast or a sling, really depends upon what bone you break and where you break it. But basically reduction of activity and then immobilization. But anybody that's ever broken a bone knows that once you've gone through that initial healing stage, what is actually recommended is that you start moving around. For example, I crushed my fourth lumbar in a car accident when I was 17. And, of course, I had to go through a period of time where I was on bed rest and, had to use a brace. But, then my doctor made me get up and start walking, because if I don't do that, then my bone will not repair itself. It won't start going through mitosis to repair those osteocytes. All right, postmortem interval. So that's the period of time between death and essentially when you found that person, postmortem interval. So evidence of trauma, looking at taphonomy. So the laws of burial, was this person intentionally buried? Were they buried is there anything ceremonial about it? Were they buried, that didn't have anything to do with homicide or suicide? Or were they buried quickly? Was it a homicide, or, it's a situation like that? Bones are subject to weathering, scattering, and scavengers. So you see here the pictures of birds and a leopard. So it's possible, even if a body was buried, it may be unburied by these scavengers. Other variables such as acidity of the soil, plants and root growth, scavengers and temperature, and also standards for estimating time of death. All right, so you can also reconstruct identity using forensic odontology, that's study of the teeth, especially in modern day when we have so much dental work done, where we have root canals and caps put in our teeth and braces, things like that. So you can actually say, for example, if I go missing and you guys want to figure out what happened to me, you could actually, and you found remains that you thought might be me, you could go to my dentist and get my dental records, and that's probably the easiest way that you could determine whether those remains are actually me, okay? All right, so also looking at if somebody's had, in modern day, if somebody's had plastic surgery, so you may be able to also identify, this is actually about reconstruction. So this is the 3D reconstruction, because forensic anthropologists understand tissue depth, how deep the tissue goes, depending upon some various factors. So again, if you had, say, a picture of me or the picture of the person that you were looking for, you can use this knowledge of tissue depth and how it attaches to the skull, how the muscles in the face are oriented, to reconstruct what that person would have looked like. So if you just have the skull, you're using this information to reconstruct a picture or an image of what she would have looked like or he would have looked like. Okay, so what do we do with this information? So we're taking the chain of evidence. So forensic anthropologists are oftentimes called upon in court cases. I'm sure, obviously in many TV shows and movies, forensic files, I'm sure you've seen this, forensic anthropologists and bioarchaeologists sometimes get called upon as expert witnesses, because they're the experts here. They're able to tell you, this is why I think this person had this biological profile, or this is why I think this person died not of a natural cause, but from blunt force trauma, for example, or this person was suffocated. There's a bone in your neck called the hyoid bone that oftentimes breaks if you're suffocated. So, then nobody can explain that better than an expert. So forensic anthropologists sometimes spend a lot of time in court if they spend a lot of time identifying the victims of violent crimes. Okay, you guys have questions? Wow, I'm proud of myself. I did that in exactly half an hour. Did I go too fast? You guys have questions about things? You guys are by far my quietest Zoom class. Are you guys doing all right out there? You guys okay?
I'm doing good.
All right.
You guys have any questions at all?
So just to clarify for, you said we're doing the conceptual questions for Lab 7?
Correct, yeah. So unit 2, we're in lab 5, 6, and 7. So you should do the concept review questions for 5, 6, and 7. Most of you have already done 5 and 6, but if you haven't, you still have another day. 5 and 6 are definitely due no later than tomorrow at midnight. Lab number 7, you only need to do the concept review questions sometime before next Friday, and you should do the lab exercise that is related to sexing the pelvis, and the lab exercise that is related to determining the manner and cause of death. So I'm going to go through right when I get off the Zoom. I'm going to go through and label them when you're in, when you're in the modules. I'm going to label the exercises. 2 of them will be labeled as required, and the others will be labeled as extra credit. Okay. Now for lab 5 and 6, we've already been working on for 2 weeks. I do want you to do all the exercises for 5 and 6 because 5 and 6 is super important, guys. Not only obviously because you're going to get points for it, but understanding the human skeleton, understanding how the bones articulate, understanding the features of the bone, especially for the skull, femur, and pelvis, those 3 main aspects. It's going to be, learning it now is going to make your life easier the whole rest of the semester. Okay. Does that sound like a fair deal?
Awesome, yeah, thank you.
Awesome. So yeah, 5 and 6 do everything. 7 is basically just concept review, and there's going to be 2 exercises from 7 that'll have you do. The rest of it's extra credit. Okay, now this is kind of a shorter unit. Next week is essentially the end of our unit. So if you haven't started, definitely get started. Don't let the same thing happen that happened last time, because I know most of you are really good. I'm not giving you guys a hard time. But there were a few of you that decided to wait till the very end before you start submitting work. And remember, the negative thing there, if you wait till the day before it's due or the day that it's due, is then that's kind of taking away your second attempt. You're allowed 2 attempts on all these exercises. I go through and I manually grade all the short answer questions. Canvas will grade the multiple choice, and I go through and manually grade the short answers. And I provide you feedback. I type in feedback if there's an area where I want you to improve, or if there's an area I want you to check out the book again. But the only way you can benefit from that is if you start submitting these exercises and get them in at least a couple days before the deadline. Okay, because I don't want you to end up not being able to take advantage of your second attempt. So next week, if you recall, I'd asked you guys last week, I have a doctor's appointment that I've been trying to schedule for quite some time now, and they had a cancellation. They can get me in next week at 2 o'clock. But obviously that would make it kind of close to be back here for a Zoom by 3. Are you guys still okay with meeting next week from 4 to 5 instead of from 3 to 4? Is that still okay with everybody for next week?
Yeah.
Okay. All right. So just like normal though, guys, if you set aside 3 to 4 and you can't do 4 to 5, I will post a recording. It's been taking a couple days for Zoom to make it available. So just keep in mind, if you're planning on, not attending and looking at the recording, just make sure you set aside some time. I'll try to get it up by Friday afternoon, but try to set aside some time, because our Zoom next week will be when we're asking questions about the exam. So, if you can be here, I definitely recommend it, especially if you have questions about the exam, because our exam for Unit 2 is due not this Sunday, but a week from Sunday. Let me look at the calendar. So exam number 2 is going to be due by October 25th. Okay. You guys have other questions for me, or anything you want to go over again as far as the human skeleton?
Yeah, just real quick, just to make sure, because I'm looking at lab 6 and 5, and it says that they're due October 16th.
Yeah, the exam, the lab, well, oh, did I look at the calendar wrong? I guess I did. Okay, so yeah, sorry about that. Okay, so October 16th, that's the absolute deadline for you to submit lab 5 and 6, and for you to get in the 2 exercises for lab 7 and the concept review question. So basically the work for unit 2 is due no later than a week from tomorrow. And then the exam is due 2 days later. The exam is due on October 18th.
Okay, just want to make sure I
Kind
got the date right.
Yeah, sorry about that. We'll kind of play it by ear, guys. I mean, I would like to stay on schedule as much as possible, especially since, you know, I'm removing a lot of the work from Lab 7 from you guys. I would like to try to stay on schedule because I want to make sure we've got plenty of time for Unit 3 and 4, because oftentimes that's the best part of the semester. That's when we get to go into primate behavior and human evolution. That's generally the most fun. At least I may be biased, but in my opinion, that's the really fun part of the semester. So I want you guys to start plugging away at those exercises, but, you know, if we find that we're really overwhelmed, you know, I might be able to extend it a couple days, but let's try to stay on track as much as we can, especially since I made a lot of it extra credit. Does that sound like a good deal?
Definitely.
Awesome. Okay, any other questions? You guys are feeling good? Okay, I mean, other thing too, guys, I did send out that survey about when you guys want me available for office hours. And out of the, you know, I sent it out to all of my classes, and out of the 40 students that responded, most of you said that you would like me to be available at a certain time for office hours, and most of you said Wednesdays from 3 to 5. So I've been doing that the last 2 weeks, and I've been sending out the link via Remind. I haven't had anybody show up the last couple weeks, so I'm just kind of checking in. Is that something that you still think would be useful, or are you guys doing okay without office hours? What are your guys' thoughts? Okay, I mean, maybe just think about it a little bit, because I'm happy to be available for you guys for scheduled office hours. But it is kind of sad when I just sit here on Zoom looking at myself. I mean, I try to, you know, work on stuff when I can, but if I don't move stuff around, then it times me out. So, you know, kind of think about it. If it's something that, you know, you think would be helpful, you know, maybe shoot me an email and let me know. And then maybe in that email, let me know what your availability is. Like maybe I need to move the time, you know, maybe that survey wasn't super accurate. You know, if that's something that you would like me to be available for, you know, try to think about when would be the best time for me to be available. Okay, so you guys have a great, fantastic week. So just remember, next week, our time is going to be 4 o'clock to 5 o'clock instead of 3 o'clock to 4 o'clock. So anybody that wasn't here, there's, I think, what, 14 of you guys here today. So if you have a lab mate or a friend that you know isn't here, just make sure that do me a favor and make sure you let them know that next week we're going to be meeting at 4 o'clock instead of 3 o'clock. And other than that, just start plugging away at those exercises and shoot me an email if you have questions. You guys have a fantastic weekend.
Thanks, Professor. Take care.
You're very welcome. You too.